I'm a grad student in Debbie Mark's lab, like Liz mentioned, and last spring, I guess, Adam, Debbie, and I came to MIA and we told you about the kind of work that our lab is mostly known for, namely building generative models, probabilistic models of genetic variation, and trying to use that kind of modeling to do interesting things like predict protein structure. But today I want to tell you about something pretty different from all the kind of stuff we've published in the past, and this project we've been working on for the past couple years or so in a pretty different direction, still on the same problem of protein structure. That's also by the three of us. And I think I'm particularly excited to present it at MIA because I think the kind of approach that I'd like to tell you about today is really about how we combine the kind of classic models and algorithms that we're used to using in different fields that we might study in computational biology and combine them with new kind of deep learning approaches that can really handle high dimensional data and large data sets in very flexible ways. I think this mic may be a little high, but. Great, so without further ado, the protein folding problem. It's hard to overstate the importance of it. You all know that turning sequence information into structure information is important, but it's always good to be reminded. If we know how to turn protein sequences into 3D structures, we can do lots of useful things. For instance, designing molecular tools that can be used in genome engineering or synthetic metabolism. Understanding protein structure is really important to interpreting drug targets and designing potentially new biologics. Of course, classically, understanding protein 3D structure is an important component of understanding biology and disease mechanism. And lastly, if we really knew this map from protein sequence to structure very well, then we could potentially do some crazy things like potentially design new protein materials from scratch that can do functions that we've never been able to do before. So the classic way that we turn sequence into structure is with experiments. And experiments are great, and they've told us almost everything we know about structure, and yet they are very expensive. So experimental approaches tend to require both big machines, experts, and money. Well, the big machines being, for instance, like maybe a synchrotron light source or an NMR spectrometer, or the kind of easiest, cheapest version, an electron microscope. These are all non-trivial barriers to entry, and so whenever we use experimental structural biology approaches, we have to be very careful with how we allocate these resources on what kind of structure problems we pursue. As a consequence, we're limited in how much of the structure universe we can peer at. And at the same time that all these developments have been going on, there's been a lot of developments in genome sequencing, which shows us how much of the kind of protein universe is out there to see in the first place. And unfortunately, what these genome sequencing projects are reminding us is that there's a lot of protein diversity out there, and our experimental structure determination efforts are kind of showing us maybe only about half of it. So in this case, this plot right here is showing you a kind of overview of PFAM, the protein family database. If you've ever annotated a metagenome or something, you might use this. Essentially, we take all the genomes out there, we find similar proteins, put them into families, and there's about 16,000 of them right now. That have anywhere from tens of sequences in them to hundreds of thousands. And if you plot all of them out, every single point here is an entire protein family. You find that about half of them do not have any of their members having an associated structure in the protein data bank. And that includes families that have lots of sequences, little sequences, and are made of small proteins and large proteins. So there's a lot of the structure universe left, just of the things that we've already seen out in the wild. So, of course, the other axis of trying to understand protein structure that's been around for a long time is computational approaches, and that's what I'm going to talk to you about today. This is not a new idea, and for many decades, people have thought of lots of different methods to take, let's say, structure information or knowledge of physics that's out there and turn it into predicted protein structures for new sequences that we see. And while this is probably could be the subject of its own 1-hour AMIA seminar, I'm going to make a gross oversimplification and tell you about the kind of two main camps of modern methods today that are maybe the state of the art that we use in protein structure prediction with computers. The first of which is going to be what the physicists do, and the second of which is going to be kind of what the statisticians and machine learners do. So first, the physicists like to combine their knowledge of physics expressed in terms of force fields that try to approximate the effective physical forces governing polypeptide folding. All you need is one sequence, this information about a force field, and then some big, powerful hardware to try to just brute force simulate the process as it goes on. And what we see with these kinds of approaches, even when you use really sophisticated supercomputers, let's say like Anton, produced by D.E. Shaw Research, that these approaches can basically fold small proteins some of the time. On the other hand, what I told you about last time that our lab was at MIA is there's this other side of the spectrum where we're not trying to do everything from the kind of bottom up, but where we say maybe we can find useful information out there in large biological databases that can inform us about structures, so much so that we can figure them out much faster. So I'm kind of loosely describing this as statistical restraints, but one of the dominant methods here are the methods based on the notion of coevolution. So the idea is that if you look at sequence variation, so you have many copies of a protein that you find across genomes, and you now ask the question the right kind of way of which columns of this multiple sequence alignment are covariant with each other. And if you use the right kind of model, you can see the last MIA seminar to hear a little bit more about that. If you use the right kind of model and you quantify what the strongest directly co-evolving pairs are, and then now you compare the strongest co-evolving positions in the protein with a 2D map of the contacting positions in the protein. So you stretch the protein out on I and J and then put a gray dot wherever there's structure. Oh, that's the wrong button. The drama is the orange lands on the gray. And this happens with such precision that you can actually often fold up structures using basically just this information. So that was the second of the two kind of dominant methods people use to fold proteins. And what you'll see about these methods is that there's a trade-off. On the one hand, there's physical approaches. They're very general in the sense that we kind of think the laws of physics, as long as we're writing them down correctly and simulating them correctly, apply in all scenarios. So we only need the sequence and our knowledge of physics. On the other hand, we need supercomputers to fold small proteins some of the time. When we have statistical information, we can often create models of protein structure really fast, but it requires that we have some other information source. So it's less general. We need external information, and we can't apply this to all the protein families, or even to out in sequence space. So an ongoing question is, how can we kind of get the complementary benefits of physical approaches together with the speed and efficiency of statistical approaches? And if you kind of take this trade-off and make a cartoon plot, you might think of all the methods that exist, whether talking about molecular dynamics, coevolution, or something I didn't mention, but you may have heard of called homology modeling. These are all kind of methods used all the time to predict protein structure. If you plot essentially how much external data do I need, or you might also think of it as how many families will this be applicable to, from molecular dynamics needs little sequence. Homology modeling needs homologous structures, perhaps the most expensive requirement. And essentially, this is a generality efficiency trade-off, and we want to get out here. to be able to push any of these methods, let's say to make MD faster, or to make these kinds of methods in the bottom right more general. And so this is kind of giving you a very brief taste of the kind of trade-offs and where the methods live in the field right now. But today, I also want to frame what I'm going to talk about slightly differently as something that might be closer to what we talk about at MIA, which is how do we build genotype-phenotype models for really complicated, potentially high-dimensional structured phenotypes. So this is just rebranding the sequence structure problem as a genotype-phenotype problem, which might be reasonable. And the challenge is that, like, of course, this is not going to be modeled by anything like a linear model or genetic architecture. The types of dependencies that the atomic coordinates have with respect to each other is very complex and is very high-dimensional. And the process by which a sequence is turned into a structure seems even hard to conceptualize as a single kind of probabilistic map. But if we had this map, it'd be really useful. And I think today I'd really like to talk about how can we build these kinds of maps. And it might, today is all about protein folding, but you know, this is true in other areas, whether we're talking about developmental biology. How does kind of genetic changes lead to kind of large-scale morphological patterns, or even in like population genetics, so it's not a genotype-phenotype problem, but we often have to learn complicated mappings, and how can we build them and be informed, especially by mechanism. So getting to what we're actually talking about, today I want to tell you about trying to build probabilistic machines that can learn to go from sequence to structure. And zooming out a little bit, I want to frame that more in terms of how can we use classic things, let's say like simulators, as core components that we can combine with deep learning models that we might use as we try to enter biology. Simulators, I think, are really important because they basically are a way to express our knowledge of how a system works. And while I don't think they're particularly utilized as in hybrid form with models, it's actually not so hard or unfathomable to start combining them. Then I want to take this idea very seriously to build an end-to-end differentiable protein folding engine. That's the kind of main event. This is actually, there are some real potentially fundamental challenges with trying to use end-to-end differentiable approaches with long simulations. And if there's time, I'd like to tell you a little bit about that and some of the kind of tricks that we've developed that can potentially get around that. But okay, let's talk about simulators. So in the protein folding problem, the kind of essential simulation process, if you will, that's happening is built all around energetic optimization. So kind of this is true for all the methods I told you about earlier, more or less. You get an energy landscape. So here, if you've ever seen an energy funnel, maybe you've seen a cartoon like this. It's not actually what's going on, but you kind of think of the location on the plane as a position in structure space, and the vertical is like the energy or the favorability. Everybody thinks both theoretically and in terms of protein structure prediction that the protein folding process is about kind of going from unfavorable high energy states down to low energy states, and this is what gives you useful structures. And this energy landscape can come from lots of places. So for instance, the coevolution methods that we use get added to, let's say, an otherwise physical energy landscape. This could just be straight physics, and also you can frame homology modeling as an energy landscape problem. And once you have an energy landscape, most protein folding pipelines are built around trying to find the ground state quickly. So that includes lots of different methods. Maybe you've heard of them, for instance, molecular dynamics or simulated annealing, or even replica exchange. So if protein folding is kind of driven internally, or protein structure prediction in general, is this simulator-like process where you're going downhill on an energy landscape, this can also help inform of all the places in which it fails. So what often goes on when these protein folding algorithms don't work is that we don't run our simulators long enough, or maybe there isn't even going to be enough practical time in the universe that they're ever going to get there. But let's say you download some molecular dynamics package, run it on your laptop, and try to fold up a medium-sized protein. You're not going to get a folded protein out. You're going to get some kind of high-energy unfolded state. That's not what we want. And this has to do with something kind of fundamental with how this field has developed. Basically, we learn the energy landscapes kind of independently of the simulators that operate on them. And today, I'd like to suggest an alternative approach where we try to learn them together. So kind of all protein folding pipelines look like this. So you take a sequence, and you have some notion of energy and some kind of simulation or optimization process, and a structure comes out the other side. But since this whole process is just a big pile of computation, and we have good paired data of sequences and structures, why couldn't we build this entire machine as some kind of differentiable engine and use automatic differentiation, or maybe you know about it from machine learning as backpropagation, to train this entire process end to end. So to get a better sense of how these types of energy landscapes might be thought of as simulators, let's start drawing cartoons that might be more familiar. So you might,
is there a tension at all between sort of the minimal energy landscape or crystalline structure and the one that might be the relevant one? Sort of that might not always be the same, and the coevolution methods might get at more of the, like, what's sort of relevant functionally and blah, blah, blah.
Yes, so I think Alex is asking a question, so we don't have mics, so I'm going to repeat everything, about how sometimes there are these methods that if you train on crystal structures, but crystal structures are perhaps not representative of in vivo sometimes because they've been forced to crystallize, then you've baked that into any type of model that you train on crystal structures, and it's going to be there. On the other hand, if you use coevolution methods, they don't have any knowledge of crystal structure. They just care about what the protein has done throughout evolutionary history. That's a very real trade-off, yeah. It seems like the hope is that the artifacts that you get are kind of not so systematic that when you train on natural structures that they somewhat cancel each other out, but you're definitely going to probably see things over-compacted, et cetera, like that. It's a very real trade-off. First, before we get into training simulators on data, it's helpful to think about how can these become differentiable machines in the first place. So let's draw a simulator more like a neural network. Well, you notice the simulator takes in a state, let's say coordinates at time 0, and then it operates on it a few times with some kind of inner loop time stepping function, and then outputs the state that you get at time t. Way to look at this more like a neural network is to unroll it in time. So instead of thinking about this as something with loops, we can think about it as a fixed computational graph that starts with the initial coordinates and keeps passing them through the same function again and again and again. And this function is powered by parameters, and maybe there's some other inputs that happen throughout time. And if you draw things this way, a simulator doesn't look that much different than, let's say, like a neural network that operates on sequences called a recurrent neural network. So if you're familiar with those, you can see some parallels. In both cases, we have parameters that govern the time stepping. In RNN, those would be the weights of the RNN cell. In a simulator, these would be the energetic restraints that govern how the physics is determining how you update the structure at each time step. The state in our simulator's coordinates, the state in RNN is the hidden unit activations, and then you might also have inputs that occur throughout time. And the reason I'm drawing this analogy in the first place is because if you think about it this way, it becomes not so unbelievable that you could train a simulator much the same way that we train neural networks. So how do we train recurrent neural networks? Well, we use backpropagation through time. This is just a special form of reverse mode automatic differentiation. And the important thing to know is that it's cheap. So backpropagation through time essentially allows us to, first, we compute our simulation. We just run it forwards in time. and then we produce an output. And now let's say that we want to train this simulator to make this output have some attributes. Well, one way we could encode that requirement is with a loss function that takes in this final output, criticizes it in some way, and now our goal is essentially to tune, let's say, the parameters of the simulator to optimally minimize a loss function. And backpropagation through time basically says the amount of compute you spent to do the forwards pass and run the whole simulation forwards in time is basically the amount of compute you need to do a backwards pass, where as you march backwards through time, you now compute as you go along the sensitivity of the final loss with respect to all the intermediate things that you encounter along the way. And so this is basically the training algorithm for recurrent neural networks in general, and you can see it would also work for simulators. One small point that's not particularly important, but to follow the details of, but I think is interesting, is that you can even have stochastic variables in here. So imagine, for instance, that these inputs at different times were noise. This happens whenever we have, let's say, noisy simulators or like stochastic differential equations or processes that maybe can use random noise to explore. And there's this great thing that underlies a lot of machine learning models, notably VAEs, variational autoencoders, called the reparameterization trick. And stated a few different ways, you might just think of it as saying that it turns out that if we have a noisy process where these were random variables that inject noise into our simulator over time, then we can compute the gradient of the average behavior of our simulator, the gradient of the loss of the average behavior, simply by sampling random values a bunch of different times and then computing the gradient, just treating those as constant. And so what this means is that, and if you want to learn more about that, you can read about variational autoencoders and other kind of stochastic computation graph literature. The important thing is just that you can even use backpropagation to optimize noisy simulators. So, okay, well, that was the theory of all this, and now we're going to get much more concrete. So can you learn by simulation? First, let's do a toy model. Let's say that we want to have a simple energy landscape, perhaps something that just criticizes the structure based on distance restraints. We're going to have no notion of sequence here. This is just a simple geometry problem. And we have a simulator, and it's going to spit out a structure, and we would like to make that structure look like some target set of coordinates. So I just pulled these off of a small protein domain. You can look at the plot of the 3D coordinates here, or a distance matrix like this. And then now let's say we just spit out a structure, and then we compare with some loss function that basically measures how similar are the two distances. Our energy is going to be distance restraints, so fleshing that idea out a bit more. It's just a sum of pairwise distance constraints. So if you've ever seen kind of in chemistry, you often describe the favorability of two things being close in terms of potential wells. And so these are just objects that say, in this case, that xi and xj don't like to be too close. They like to be just this distance apart with some sloppiness, and then if they're completely dissociated, it stops mattering at some point, but you pay an energetic price to be dissociated. And then we run a simulation. I'm not going to go into the details of that, but it's essentially just a simulation where you're kind of going downhill with momentum and noise. But now let's see what happens. Okay, so start. We have initially randomized simulator. So every single one of those ij distance restraints is set to something kind of simple, and it has no knowledge of the target structure. And the output it gives us at the end of the simulation is some elongated thing that doesn't look at all like our target. Let's start learning. So now the movie we're seeing is every single frame of the movie is a training step. And every single training step is an entire simulation. So every single frame of this movie was an entire simulation. You can see it, for instance, up here. So the simulation that are happening in each frame start with an elongated, something that looks nothing like the target structure. And then the simulation itself that happens within each frame is using the energy landscape, and it's going downhill, and it's noisy, so you see it's going kind of all over the place. But what you can see, this is looping, is that as it goes through, slowly but surely, the simulator is able to learn how to manipulate its energy landscape such that this process that's happening at each frame of the movie ends up with these target coordinates. And where it gets, I think, the most interesting is to actually see what's happening to the energy landscape itself. So if you watch here real fast, it'll reboot in a second. So watch the diagonal. You'll see that essentially it learns short range to long range. This is the distance matrix. So right here, these are essentially every ij between two, let's say, positions in this chain has some associated potential well. That potential well, as I showed you, has three parameters, and you're seeing just how these parameters evolve over learning. So you see it can basically, of course, this is kind of in some sense, the solution is not so surprising. It just learns, it basically uses the potential well to anchor the structure around the contacts that happen in the target. So this shows, essentially, we can backpropagate through entire simulations, starting with no information about how to fold something, and we can learn how to fold stuff up. Okay, so now,
John,
yep.
can I ask you to take us more slowly through the bottom right? Right, so we have these potential functions. We have one for each pair of atoms,
Yes.
or amino acids, rather.
So I'll reiterate it. We have potential functions, one for each pair of amino acids. So for instance, each little circle in this plot is a coordinate, x, y, z. And for every pair, then we have these. Yeah.
Okay, and then can you describe each of the three in the lower right in turn? This looks really interesting, and I want to make sure I.
Oh, sure, yeah. It was, and I definitely was going a little faster than comprehensible, hoping to get to the next part. But I think this is really where you can see interpretability. So every potential well has three parameters, one of which is basically what distance do I want to be apart, how much do I care, and how much does it cost to not be bound. So those are the three parameters that are being plotted in that matrix: length, scale, and depth. So essentially what you're seeing here is this one says, what distance would I ideally like these ij's to be apart? And it starts not really knowing anything, and then it kind of learns, memorizes the distance matrix more or less. And then this says, how much of an energetic boost do I get if I do that correctly? It's the depth of the potential well. And then finally, this one essentially is like a precision. It's like how much, if it's bigger, then it's then you pay a particular price if you are not that exact distance apart.
So floppiness for that pair.
It's the floppy, yeah, it's floppiness.
And the data that you're training on, can you say one more time briefly?
Oh, so it's not like this is not going to be any of the sequence to structure yet. Sure. It's just literally that is the data.
You just have one thing.
We just pulled a C alpha trace.
Yeah. Okay.
Yeah.
So can I just ask one more question? So the parameters you see down there, they are used throughout the whole simulation, right?
Yes, the parameters are used through the whole simulation.
And then this is the updating throughout when you train.
Yeah, this is a very tricky part about these movies that I apologize for. There's two time scales going on. There's training time, which is the movie, and then there's the actual simulation time that's going on, which is, I don't show you a movie of, but it's just give you snapshots of the beginning to the end of simulation. So this is, sometimes people might call this meta-learning, and essentially what you're seeing is how the whole simulator's parameters evolve. And then you can also see snapshots of how the simulation's evolving over time. Yep.
You just run the simulations for a fixed amount of time and then take the end structure and calculate a loss based on that?
Yes, we'd run them for a fixed amount of time, and then the hope of this whole kind of philosophy, I'd say, is that because we only give it a fixed amount of time, we're saying you have to figure out some way to use an energy function and in combination with your simulator to figure out how to make this happen. And so that's like, now we're forcing the energy landscape to really produce a useful answer. And
is just your energy function parameterized, or do you parameterize your simulator as well?
Right here, we only parameterize the energy function, but later in the big production model that I'll tell you about in a bit, we parameterize both the energy function and the simulator so they can kind of co-adapt. Yep.
So it looks like your parameterized energy function is converging, but have you tried running this over several different trajectories just to see if you get the same representation of Morse length, depth, and scale each time?
I have not tried repeating this experiment very thoroughly. I've done it maybe like a handful, 2 or 3 times, and anecdotally, I think it seems to always have roughly this outcome where it just memorizes the distance matrix, has some long-range depth, but I don't know anything quantitative in terms of how reproducible, let's say, the particular details are.
Do you know of any other examples of basically doing backpropagation on a model where the parameters are all fully physical and interpretable? I mean, this seems like kind of a first.
I'm not so sure of many examples of backpropagation through simulators where the end goal is going to be to like bring this together with neural networks. Classically, there's this whole subfield of let's say industrial design. Let's say like how do you design an airplane's geometry called like adjoint sensitivity analysis. And it turns out that this method is basically just a rebrand or the earlier version of something that's closely related to backpropagation. And so, for instance, the way people design optimally non-turbulent airframes and stuff is, as a goal to get interpretability, there may be some examples I'd like to see more. Yeah. But that's so Alex's question, I think, is great because the hope with these types of approaches is that by putting simulators inside of the models that we build of biological data, we're going to be able to interpret what's going on in the frame that we kind of have classically understood the system. Yeah? Yeah, sure. Oh, sorry.
Can you talk about the loss function that you use to keep sides back and how you choose it?
So the question was about the loss function. I think in the interest of time, I will not go into it too much other than saying that it was just comparing the pairwise distances. One way to do it is by subtracting them and squaring them. Another way to do it might be to take log ratios. It turns out that using log ratios is a little more natural in the sense that short-range things that change should be more important than long-range things that change. So if I'm 82 versus 2 angstroms apart, that is much more important. That is much less important than if I'm 2 or 4 angstroms apart. So doing everything with kind of log differences rather than linear differences ends up being, I think, a more natural way to do it. But I'll talk about another loss function later, and I just want to make sure I get there. So that's about as much. Yeah.
Is it only the final time step that goes into the loss function?
For this one, it is only the final time step that goes in the loss. Later, it's everything. Okay, so where are we going with this? Well, earlier I just showed you something that did not have a sequence on the left and a structure on the right. But we want to build something where a sequence goes in on the left, a structure goes out on the right, and some type of complicated computation, potentially interpretable, occurs on the inside that we can fit to potentially large data sets. And this is what we've come up with. It's a neural folding engine. So I don't show any of the neural networks here, and I won't talk about them too much today because they're not so different from a lot of the things people are doing in the literature. But roughly, there's 3 components here. There's a sequence that is then what an energy neural network takes in, and then what it does is parameterize all of these terms in an energy function that essentially says what does it think this protein's conformational favorability should look like. And then this energy network interacts with a simulator that attempts to draw a low energy sample from the energy landscape. Something that's important is that this is going to be coarse-grained. I'll mention that in a second. And lastly, this coarse-grained model is turned into an atomic model, and now you have sequence going in and a structure going out. But now let me tell you a little bit about some of the details. So first, the energy network. Well, I mentioned to understand an energy network, you have to understand the coordinate systems we use to describe a structure. So just in the interest of computational efficiency and also simulator efficiency, our folding process is going to be coarse-grained. So just like the previous movie, each amino acid in the protein is represented only by one coordinate. And so naturally, the way to describe a chain that you might be familiar with is Cartesian coordinates. So we have kind of 3 points, normally named x, y, z, that would be associated with each bead in a chain. It's important to note that you can also define an internal coordinate system that you can invertibly map back and forth between Cartesian coordinates. Internal coordinates are often much more natural for molecular geometry problems. We instead describe each coordinate, let's say xi, as in reference to the preceding coordinates in terms of basically planar dihedral angles, bond angles, and bond lengths that tell us essentially in the reference frame of these beads, where do we place this next one? But it's important to understand these two coordinate systems because each of them is more naturally represented in the energy function. Okay, so this is going to coarse-grain an entire neural network into a line diagram, and unfortunately there's just not time today to talk about the details. But essentially, our energy network takes in a sequence, processes it using ideas that come from both normal like image kind of processing, 2D convolutional neural networks, and also graph neural networks. These have been really powerful in chemistry. And then it outputs two shapes of information, you could say. One is 1D, so along the chain, and the other is 2D in the sense that it tells us about relationships between positions in the protein. And then each of these 1D and 2D pieces of information can now parameterize 1D and 2D restraints. So 1D restraints being angles and bonds, and 2D restraints being distance restraints. So let's look at what those terms look like. So just like before, our 2D restraints, the pair potentials, are also going to be ij, except before we just had introduced free parameters for each ij. Now these ijs are the output of a deep neural network. It's slightly more parametrically flexible than the potential I told you before, but has the same qualitative features where there's short-range repulsion, potentially long-range attraction, and then some parameters that describe essentially the location and shape of the optimal distance of something. And then for angles, this might be tricky to understand from this graphic. I don't think it really does explains it too well, but the thing to take away is that what the angle restraints do is they look at a sliding window. You can kind of think of it as a fragment of the backbone of the chain, and then they essentially use convolutional-like ideas to criticize, does that joint configuration of that fragment look happy? And you can do this kind of by lifting all the internal coordinates along that window into a feature space and then pushing it through like convolution and nonlinearity. But you can think of these as just, it knows nothing about how proteins work when it starts, but then we give it these parameters to kind of say who should be, like which positions in the protein should be close, and what should the chain look like. And we kind of really think that these two different ways of thinking about protein structure are very important and kind of different from one another. On the one hand, this kind of tells us about how things pack and go together, but it turns out that encoding precise angle information in distances is very hard and imprecise. And you can't even encode chirality at all. On the other hand, angles are much better at encoding let's say the local structure of proteins, but they're not as good at saying like which parts of the protein should be close. Okay, so that was the energy network. It then feeds into a simulator. That essentially, so the energy network also spits out the initial conditions to the simulator in terms of bond lengths and angles. And then the simulator is now going to do dynamics to try to optimize this energy function. So I can't go into too much detail of what it looks like, but I can tell you the really hard problem that kind of held us back and what we had to solve to get this working. Basically, you need to maximize the utility of the simulator budget that you have. And we find that the way to do that is when we do the optimization, we don't, the kind of motions and the steps that we take in structure spaces where optimizing it are not going to be Cartesian, they're going to be angle motions. So it turns out that, you could write the equations of motion in Cartesian space, but then essentially all the beads are wiggling around independently of each other. This can't really move anywhere fast, but if you write equations of motion in terms of this dihedral angle spinning, this angle changing, this bond extending, you get these big coherent motions where this structure is locally preserved, but it's making big moves. So kind of put some equations behind what I'm saying. The naive way you might do it would be, let's say so we've done it with a few different kinds of simulators, but Langevin dynamics are essentially gradient descent with noise. You might encounter them in physics, and they essentially, we at each time step, we would take Cartesian coordinates and go down the energy gradient a little bit, and then add some Gaussian noise in the right proportion. This is interpretable because this is both how we think proteins fold physically, though of course if you use a weird energy landscape and you don't have it tuned, let's say, to physical things, it's not that it's physical, but the point is it's sampling from the Boltzmann distribution that the energy function is associated with. And then, so this is the naive way to do it. And the thing we had to do to get this really working was rather than write the equations of motions in Cartesian space, where you're using x, you can write them in internal coordinate space. And I won't explain it too much, but you can add, this is related to a question earlier, you can add a preconditioning matrix, is sometimes called like a mass matrix, essentially something that tunes the behavior of the simulator to know which degrees of freedom should be moving faster or slower. This can all be learned. I won't go into this too much more, but this was kind of one of the big bottlenecks of the project, figuring out how to get a really fast simulator. And there was a tricky bit where it turns out if you do this equation naively, it breaks all GPUs, breaks TensorFlow, and we had to have other, let's say, equations that implicitly do this, but with a kind of different mathematical scheme. But for another time. So that's the simulator. It folds stuff up. I'll show you movies in a bit. The last part, refinement, is it's straightforward. We just take a coarse grained structure. A neural network looks at that coarse grained structure and says, well, where should the atoms go? And then the final thing that comes out is not, let's say, all of the atoms in the structure, but it's all the heavy atoms of the backbone. So that's nitrogen, 2 carbons, and an oxygen. And then also a fifth thing, which represents the kind of coarse-grained location of the side chain center of mass. And we want that data in there because kind of side chains are everything in protein folding. And if it doesn't have some idea about where those are, I think it's not necessarily going to be able to learn. So then the structure comes out the other side. How do we criticize it? Won't go into this too much. There's lots of different ways that you can compare the structure that it should be and the structure that came out of the model when it's still trying to figure out what's going on. You can use lots of different terms in LAS than we do. You can even make things that are kind of more closely related to protein folding, like hydrogen bonding and TM scores, differentiable. That's the great thing. Once you have end-to-end differentiability, you can just optimize as many things as you like. You can also there's a theoretically more correct way to do this with something related to GANs, but I can tell you it's very unstable. Okay, so in the last few minutes here, let's just think what does this thing look like learning? So as a starting point, wrong button again. As a starting point, let's just look at a simple, easy to understand data set. So the Baker lab that does computational protein design put out this paper last year. It's awesome, where they essentially designed 4 mini folds and then systematically created lots of sequences for these folds from scratch, all designed computationally, and then in a high throughput assay validated that all of them fold up. So this is nice because it's kind of, rather than going straight to the PDB, we can see what happens when you have kind of IID splittable data.
What does 4 folds mean?
So a protein fold is typically like, let's say, an arrangement of secondary structural elements in 3D. So maybe a fold in this case might mean that you have like an alpha helix and it flips on itself and then makes a beta hairpin, and that's one fold if it does it a particular way. So they have 4 kind of those like primitive shapes. So after you train the model, you're now seeing what kind of stuff does it learn to do in the coarse grain simulation. So down each column on the left, the static columns are the target that it's supposed to be predicting. On the right was the way the simulator got there. And you can see the nice thing, this is test set, so it seems like it's moving stuff. This can learn to move stuff. It knew nothing about proteins, and then it invented these force fields on this simple problem. And now, but where I think you really get this is really illustrated is if you look at, let's say, what the structural output is over the course of training. So at the beginning of training, the output of the model, if you try to put them in protein folding software, looks crazy, like these crazy hairballs, where you actually have to set extra settings because the atoms are so all over the place, it doesn't make any sense. And then slowly, as it does training steps, the outputs that it predicts become closer and closer to the target. So each of these is the output of a full simulation, but this whole picture is meta. Yeah.
What are you training on?
So this is just training on the 4-fold Baker data set. Yeah. And so as you go along, you see that this ensemble converges to the target, and this is a restricted sense. But the nice thing is because it was all designed from scratch, we just do IID splits, and what you find is that this is a score that people like to be above 0.5 in protein folding, basically is good both on train and test. Right. So now the next section is going to be a fast thing that I would just call the gallery of learning, where essentially, so what's the obvious next thing to do is to just set it loose on the PDB and all the sequence databases out there, and that's really what the effort of the past year has been. But I just want to show you kind of maybe a compilation video, almost like, let's say, a high school or a scrapbook of your time in high school, as you went through trying to learn different things that first day that you could learn to unfold and then refold proteins the right way, but only 10 at a time, but at least you kind of figured out how to put things in the right place. Then we gave you more structures, and you could unfold them and refold them back together. But this isn't de novo. But it seems like you're starting to figure out some physics and interesting motions. And then on test, it seems like sometimes you're good and sometimes you're bad. And then we really go crazy and try to do everything from scratch. Again, the static columns are the target, and these are all snapshots as the model is trying to learn. And the thing you'll find about these types of approaches is that they can take a long time to converge. So today I won't be showing you, let's say, any finalized results, but you can stay tuned. They can do lots of different behaviors emerge from this kind of simulation engine as it goes through. And let's just do one last one. You can notice some colors look like they're in the right spot, some don't. But before going into any comparison results, I think this is nearing the end here. I can give you a quick vignette of the kind of okay, so yeah, building that is some work and a challenge, but there's also some deeper challenges that exist in using simulators in general. That is kind of the story of how this goes as a grad student project, let's say. Well, often you'll set off one of these models, and it will just be learning for a while, and you can measure how it's learning. In machine learning, we often look at the norm of the gradient. This is telling us how to adjust our model parameters, and then suddenly, as it's going, you'll get this crazy explosion where all the signal that you're using to learn is diverging to nonsensical numbers, and nothing makes sense. And it turns out that this is deeply related to the kind of meta thing that we're trying to do. So it turns out that instability. We're doing meta-learning here, where we're learning an optimizer and the optimization landscape all the time with optimization. So in that kind of scenario, which was explored by Dougal, McLaurin, and David Duvenot's paper in Ryan Adams' group a few years ago, when you do meta-learning, sometimes small changes in the step size that you change on the landscape can make crazy changes in how the gradient behaves. And while that might just sound like usual run-of-the-mill kind of neural network stability problems that people encounter if you've ever trained those kinds of models, it turns out there's something deep here, which is that these processes that are optimization processes on landscapes have the property that they can easily become chaotic. And so, for instance, if you have an optimization landscape here, and you're just bouncing around trying to go down the landscape with some fixed step size, one of Jeff Hinton's students back in the 90s had this beautiful thesis where he showed that you can analyze this process as a dynamical system. And it turns out as you're bouncing around here, sometimes you'll get into high curvature regions where you just kind of bounce around. And if you keep track of that bouncing around over long periods of time, you get these beautiful chaotic patterns that emerge from it. And while those look beautiful, and it seems like, well, how harmless is that? Or I mean, how harmful is that? We kind of ended up at some point on the optimization landscape. So what? It turns out that chaos itself is deeply connected to not being able to backpropagate through time. So let's just explore that idea really fast with a 1D model of what we're doing. So let's imagine our protein is one number, a particle, with a position, x, in a potential well. So this is a super simple thing, and now we're going to do vanilla gradient descent, no noise. So I'm x here, and then I just take a gradient descent step, and maybe take a gradient descent step, and a gradient descent step, and I'm just bouncing around. And the kind of step size that I'm taking is set by this alpha parameter. And let's just treat for now the landscape as fixed. So one way to look at what happens to this system as we run the simulation for long periods of time is to plot every position x that is visited by this particle as it's bouncing around. This is kind of the 1D equivalent of those attractors that we saw in the previous figure. And that's called an orbit diagram, because we're looking at essentially the orbit of where this particle is visiting in x space between a bunch of different time steps. And what you basically see if you plot for the little particle in a well example that I showed you, which is kind of representative of our whole protein folding process, if you plot that orbit diagram, you get this right here, which is a classical figure if you take a class on chaos or dynamical systems called a period doubling bifurcation. Essentially what happens, so the thing on the x-axis is the simulation time step, and the thing on the y-axis is what are the states that the particle in the well visits. You basically see for small time steps it converges to a single value, and then for slightly larger time steps it bounces back and forth between two values, and then you hit some other thing and it goes 4 and then 8. And this, it turns out this object is an extremely well-studied kind of and somewhat universal phenomena that can happen in these little 1D iterative processes. But the thing to know is that as soon, you'll notice that it's doubling, and then somehow it's black over here. So what's going over here is that here we were periodic, but maybe with a high frequency oscillation. On the other side of some bifurcation here, you descend into complete aperiodicity. Everything's bouncing around. And when you descend into complete aperiodicity, you can plot the gradient. This is the sensitivity of the final state of the simulator with respect to the initial one. This is exactly what we use to learn with backpropagation through time. And you basically see that the second that you go into chaos, you get gradients that are diverging to kind of meaningless numbers. And this is actually not so, maybe this is kind of, in the one sense, tautological, because it's essentially the way we define chaos is often by something that kind of analogous to exploding gradients. It's basically that perturbations in our system can become amplified by the system itself. So the last thing I'll leave you with is this way that we found that kind of ended up being crucial to avoiding chaos in simulators. Basically, how do you measure if a system can be chaotic? You take a system that's marching along through time, you perturb it, and you ask, well, after I perturb it with a little ball here, how far apart do these things get? And it turns out that when these when perturbations can become amplified, that's when chaos happens. Now if you just regularize this to not amplify perturbations, chaos goes away. So it seems like there are ways around this after all. So today I told you about how simulators can become important parts of models that we build in biology, how you can use this to build an end-to-end differentiable protein folding engine, and how there are some challenges to overcome there. Of course, huge thanks to my lab, and also to Chris Sander and members of the Sander lab. And with that, I'll take questions.